Thank you very much. So good afternoon, everybody. Today we'll be presenting on my PhD research project on inhibition of Calpains in triple-negative breast cancer. To begin with, I would like to start by talking about why we study breast cancer to begin with. In Canada, breast cancer accounts for 25% of all new women's cancer diagnoses every year. And of those women diagnosed with breast cancer, even though the average survival is fairly good, still about 10% will succumb to that disease within 5 years of diagnosis. And also among those with breast cancer, about 20 to 30% will develop metastatic disease. And now among those with metastatic disease, about 70 to 80% will die in 5 years. So what our lab is hypothesizing is that if we perhaps can prevent this progression from local and regional disease to metastatic, then perhaps we can ameliorate this huge increase in breast cancer mortality. And the way we do that is we're looking for molecular mechanisms and targets that can be exploited to abrogate cancer metastasis. And that's how we arrive at Calpains. Calpains are a family of 15 calcium-dependent cysteine proteases. I focus specifically in Calpain 1 and 2 because they're the ones that are ubiquitously expressed and well-studied isoforms. There are many known roles for these Calpains in cellular functions, and a few of those are known to be cancer cell intrinsic, such as cancer cell migration, invasion, cell cycle propagation, and signaling and survival and apoptosis. Calpains 1 and 2 are heterodimers of a small regulatory subunit Calpain S1 and a large catalytic subunit, either Calpain 1 or Calpain 2. These consist of PC1 and PC2 domains, which make up the active site, Calpain beta sandwich-like domain, the role of which is largely unknown, and then the PAF domains, which mediate this interaction between the small regulatory subunit and the large catalytic subunit. It's important to note here that this dimerization is necessary for the activation and for the stability of the large catalytic subunit, meaning that if there is no small regulatory subunit, it will not be active and in fact, it will degrade in the cell. The way Calpain exerts those functions in the cell is through cleavage of its protein substrates. We know that there is more than 100 protein substrates, so the exact functions of Calpain depend heavily on which substrates get cleaved, and that depends on time and location of Calpain activation. The main regulator of Calpain in that fashion is calcium, and binding calcium is what activates Calpain's proteolytic activity. And that calcium has to come either from endoplasmic reticulum or from extracellular space, because usual calcium concentration in the cell is not high enough to activate Calpains. And then once Calpain is activated, again, to reiterate, it depends on where it's activated and what it's going to do. For example, if Calpain is activated at mitochondria, there are a few known Calpain substrates there. For example, BAX and the apoptosis inducing factor. And when Calpain cleaves those, that nudges the cell towards apoptosis. For example, if Calpain is activated elsewhere, for example, cytoplasm somewhere, there are multiple other pathways that can be affected by Calpain cleavage. And just to name a couple, there is the AKT pathway, the MAP kinase pathway, and also it's worth to note that Calpain can even cleave some nuclear proteins, for example, p53 and c-MYC. Most importantly for my project, Calpain can cleave proteins at a cortical membrane. For example, when Calpain cleaves protein tyrosine phosphatase 1b or Src kinase or cortactin, that affects actin branching and polymerization. And those processes are important for formation of invadopodia and lamellipodia, which are in turn important for cell mobility. Separately, Calpain can cleave structural proteins of focal adhesions, namely talin, Src kinase again, vinculin, focal adhesion kinase, paxillin. And we postulate that it's through cleavage of these substrates is how Calpain exerts its effect on cancer cell motility. In addition to molecular evidence, there is also clinical evidence of the involvement of Calpains in the process of metastasis. For example, in this one meta-analysis study, Calpain-1 expression is correlated with changes in overall survival of cancer patients. In that, patients with high expression of Calpain-1 have a hazard ratio for overall survival at 1.73. So that means that on average, those tumors that express high levels of Calpain-1 lead to 70% worse overall survival. This can be further stratified by particular clinical pathological features, and the ones that I would like to draw attention to here are tumor depth, lymph node metastasis, and distant metastasis, which all have odds ratios in favor of Calpain-1 expressing tumors being more aggressive, more metastatic. We also have evidence of Calpain's involvement in breast cancer specifically. For example, in HER2-positive breast cancer, those tumors that express high levels of Calpain lead to worse outcomes. And in triple-negative breast cancer, those tumors that express high levels of Calpain-2 similarly have worse outcomes. And even though we have this multitude of evidence that high expression of Calpain leads to worse outcomes in cancer, nobody has been yet successful in inhibiting Calpain in clinical setting. In fact, to date, there has only been one clinical study completed that attempted to inhibit Calpain. This was the study on this drug, Blade 2660, which was tested in the context of COVID-related lung fibrosis. But in this study, this drug was unable to affect any outcome measures. And in fact, the authors didn't even publish anything on whether the drug was successful at inhibiting Calpain itself. We can assume it wasn't. So that is safe to say that to date, there isn't any effective inhibitors of Calpain that are clinically viable. However, there are many Calpain inhibitors that are marketed for in vitro use, for example. And these two fall into two major categories. That's active site-directed inhibitors and allosteric inhibitors. Among active site-directed inhibitors, one common type is peptidomimetic small molecule inhibitors that work by covalently binding the active site cysteine. One major benefit of that kind of inhibitor is they tend to be fairly effective against both Calpain-1 and Calpain-2, but because of homology of the Calpain active site residues with other cysteine proteases, this kind of inhibitor tends to also inhibit other cysteine proteases, for example, cathepsins, and in this case, also proteasome to some extent. One other type of Calpain inhibitors that is active site-directed is peptide-derived inhibitors. For example, this compound PCB1B, which is based on the primary sequence of the exon 1B of the calpastatin, which is an endogenous inhibitor of Calpain. The major benefit of inhibitors like this one is they can be made to be very specific for Calpains, but they run into other issues such as poor solubility, poor cell membrane penetration, and poor pharmacodynamics in general, pharmacokinetics. Another type of Calpain inhibitors out there is allosteric inhibitors. This kind is mostly represented by these two compounds, PD150606 and AMG 853. The mechanism of action of these two compounds is similar in that they bind in this pocket of the small regulatory subunit, which is on the outside of the dimer interface. These two compounds were discovered by high throughput screening and by in silico modeling, but both of them failed any extensive validation by other groups. So again, to summarize this, there are Calpain inhibitors out there, but they tend to be suboptimal. For that reason, our lab has mostly resorted to use genetic inhibition of Calpains for the studies. For example, in this one study by Stacy Grieve, she has shown that when using shRNA against Calpain-1, she can demonstrate that Calpain-deficient tumors in mice grow slower, and that effect can be further emphasized if she combines Calpain-1 knockdown with treatment with 17-AAG, which is an HSP90 inhibitor. And most importantly for my study here, Stacy has also shown that knocking down Calpain S1 not only reduces the tumor growth rate, but also significantly reduces the metastasis in that model compared to the control. And that too can be to some extent emphasized with a chemotherapeutic treatment. So to summarize my introduction here, we know that calpains play a role in making tumors more aggressive, more metastatic. We're not certain which calpain isoform that is, either calpain 1 or calpain 2. But we also have no clinically relevant inhibitors that we can use to exploit that role of calpains in metastasis. Therefore, aim 1 of my study is to determine which exact calpain isoform is the one we need to inhibit to elicit the antimetastatic effect. And aim 2 is to develop assays which will allow us to look for novel calpain inhibitors. To pursue aim 1, we use MDA-MB231 triple negative breast cancer cells as our model system. The reason being that these cells grow into tumors that are highly aggressive and very metastatic. And we've used CRISPR-Cas9 to knock out individually Calpain-1, Calpain-2, and Calpain S1 genes out of these cells. And then we used lentiviral rescue add-backs to reintroduce that gene expression back into these cells. The idea here is that we want to see a phenotype that will be deficient in the knockouts, but then comes back with a rescue. That's a validation step. By immunoblotting, you can see that our knockouts completely abolish the expression of Calpain-1, Calpain-2, and Calpain S1. And it's important to note here that when we lose the expression of Calpain S1, we also lose expression of Calpain-1 and Calpain-2. That goes back to what I said previously, in that in the absence of Calpain S1, Calpain-1 and Calpain-2 lose stability and are degraded in the cell. Rather than looking just at the expression of calpains, we can also look at their activity by Casey enzymography. So here, white bands represent active calpain, and those two disappear in the knockouts. And again, note that in Calpain S1 knockout, both Calpain-1 and Calpain-2 activities are abolished. Separately, it's important to note that all these genetic alterations that we've done to these cells have not affected their proliferative capabilities. The first phenotype that we looked at is the cell's ability to migrate on a 2D substrate. So here in the video on the left, you can see intact MDA-MB231 cells. You can see them extending lamellipodia, migrating. In Calpain S1 knockout cells, though, you can see that many of them stay mostly static, but perhaps more interesting is the phenotype of cells like this one, which even though it doesn't move laterally, it is extending that lamellipodial front, but is unable to detach from the substrate, which results in this spinning top type phenotype. And then in Calpain S1 rescue, that migration phenotype is largely rescued. Quantify that, I've traced the trajectories of these moving cells, and you can immediately appreciate that in Calpain-1, Calpain-2, and in Calpain S1 knockout, those trajectories are significantly stunted, but it is rescued in all the rescues. There's two measures to quantify this effect. One is to look at directionality ratio, which is the measure of how much the cells are able to maintain the direction they're migrating in. And you can see that that drops off in all the knockouts faster than it does in the controls. And the other measure is the mean square displacement, which is a measure of how much area the cells have actually covered when migrating. Again, you can see the effects of the knockout. And again, to reiterate, those phenotypes are rescuable with the add-back of Calpain-1, Calpain-2, Calpain S1. This suggests that there is non-redundant roles for Calpain-1 and Calpain-2 in cell migration. So it seems that in this regard, deficiency in either calpain can have an effect. Separately, Danny and Jenny in our lab have done an experiment where they assessed invasive potential of calpain-deficient cells. And what you can see here is fluorescent images where blue is cell nuclei, and green is a DQ collagen extracellular matrix-like substrate, which acquires that fluorescence when it is digested by the cells trying to invade through it. That green signal is largely lost in Calpain-2 knockout and comes back with Calpain-2 rest. It's interesting to note, though, that this phenotype is only present in Calpain-2 knockout and in Calpain-S1 knockout cells, not in Calpain-1 knockout. So this suggests that, at least in terms of invasion here, Calpain-2 is the isoform that is important for this role. Next, to actually see the effect of Calpain deficiency in metastasis, I performed this in vivo xenograft experiment. In this set of experiments, I made GFP-positive cancer cells, which I have engrafted into a mammary fat pad of immunodeficient mice. I let those tumors grow to 600 cubic millimeters, at which point the tumors were resected. And after another couple of weeks, I performed necropsy. I harvested the lungs, and I looked at and quantified metastasis. So for this, I have 3 sets of data. For each set of data, I have 3 measures. Tumor growth curves, the tumor growth rate estimated from those curves, and the quantification of metastasis. For the first set, Calpain-1 knockout and rescue, you can see that there is some statistically significant differences in tumor growth rate. But I would suggest that even though this difference is statistically significant, it's not biologically significant. Similarly, there is statistically significant difference in the metastasis from control to knockout, but it's not readily rescuable. Similarly, in Calpain-2 knockout and rescue, we see some statistical differences in tumor growth rate in metastasis, but it's not a clean knockout rescue effect. However, when we knock out Calpain-S1, so again, that's disrupting activity of both Calpain-1 and Calpain-2, we see some difference in tumor growth and a very large difference in metastasis, which is pretty much abolished in the knockout and brought back by reintroduction of Calpain-S1. So this suggests that, at least in terms of metastasis, we need to inhibit both Calpain-1 and Calpain-2 to achieve this effective abrogation of metastasis. To visualize this effect, here is pictures of lungs from tumor-bearing mice that had either control tumors, Calpain-S1 knockout tumors, or Calpain-S1 rescue tumors. The white hyperintense nodules are individual metastases here, which you can see are gone in Calpain-S1 knockout and come back with Calpain-S1 rescue. So this pretty much summarizes the phenotype of Calpain deficient cells in terms of metastasis. And next step from here would be to try to phenocopy this effect pharmacologically. So that leads me to my aim 2, is to develop assays to conduct drug discovery and evaluate novel Calpain inhibitors. In our search for Calpain inhibitors, we decided not to go for the active site of Calpain, because historically, even those inhibitors can be made to be very good in terms of inhibiting Calpain, they're rarely specific enough. However, we do know that binding of small regulatory subunit to the catalytic subunit is necessary for its activity and for its stability. So we hypothesize that if we can disrupt this interaction between the path domains, then perhaps we can kill the activity of the enzyme. We, however, would then need a tool to be able to measure that interaction. And that's how we arrive at the split nanoluciferase system. The way split nanoluciferase works is you start with your proteins of interest. In our case, it's path domains of Calpain-S1, Calpain-1, and Calpain-2. And you express them as fusion proteins with large bit and small bit of the split nanoluciferase system. When your proteins of interest interact, that reconstitutes the luciferase, and that's detectable as luminescent output from your reaction. However, if you were to introduce some agent that disrupts this interaction, that would also result in the reduction of the luminescent signal, and that's how you would detect that you've inhibited the interaction. This system, because it depends on making these fusion proteins, is somewhat dependent on how you express these fusion proteins. Which of your proteins of interest is made as a large bit fusion versus small bit, and whether these are made as N-terminal or C-terminal fusions. Because of that, the first set of experiments I did with the help of Dr. Kazem Nouri, some of you might know him. And what we did is that we expressed Calpain-1, Calpain-1, and Calpain-2 fusion proteins in all the possible combinations. So either with large bit or small bit, with on N-terminus or C-terminus of our protein of interest. And then we made all the possible combinations of those. Here we noted that we achieved the best signal with this combination, which was large bit N-terminally fused to Calpain-1 and small bit N-terminally fused to Calpain-1. And then for consistency's sake, we also started working with the Calpain-2 biosensor, which was in the same orientation. So large bit Calpain-1, small bit Calpain-2. From here, we moved on to the bacterial expression system. And the final constructs that we were expressing were as such. Again, N-terminal large bit on Calpain-1, then small bit on Calpain-1 or Calpain-2. But we also had added histidine tags and a maltose binding protein, which were to act as affinity purification tags. From here, we expressed and purified those proteins. We expressed them in the E. coli system. Here you can see that they expressed very well, all three of these proteins. Then we affinity purified them. And the purpose for that was because with purified proteins, we can very accurately establish the concentration of our construct. And that helps us to work with these proteins in exact stoichiometric amounts that we want to. From here, I went on to do a few optimization experiments with the biosensor. The first of them was just to look at Calpain-1 and Calpain-2 biosensors in comparison to one another. And the first thing we noted was that Calpain-1 biosensor was giving us a much stronger signal than Calpain-2 biosensor was. So that's Calpain-1 biosensor, which is large bit Calpain-1 plus small bit Calpain-1, and Calpain-2 is Calpain-1 plus Calpain-2. This was a pleasant finding for us in a way, because there is other tangential evidence in the literature that suggests that the dimerization of Calpain-1 is stronger than that of Calpain-2. From here, I've shown that our biosensor assays are very sensitive, in that even at 5 nanomolar concentration of our biosensor halves, we could still see difference between our positive and negative controls and be able to reliably detect that. Next step of optimization was to study the effect of pre-incubation times on the binding of our proteins and on the biosensor assay. Here, what I established is that pre-incubation is necessary to see optimal signal, in that we observe more binding as the proteins are incubating together, but that plateaus at roughly 60 minutes of incubation time. Next, I looked at moving on from 1-to-1 stoichiometric ratio of our biosensors, and I've shown that when we change that ratio to 10-to-1 either way, we're able to yet again increase the strength of our biosensor signal. But depending which way you switch that ratio around, that would affect our signal to noise ratio. And the final optimization step that I did was that I've shown that regardless of the incubation time of our proteins in the assay conditions, even up to 6 hours, our proteins were not degrading, the signal strength wasn't degrading. So that just goes to show that our assay was robust for any other screening or test purposes that we would need to use it for. After the optimization steps, I did a few validation experiments. Validation, what I mean by that in this case is I wanted to verify that that the results we get with the Calpain biosensors are representative of the endogenous structure function of Calpain-1 and Calpain-2. And separately also wanted to make sure that we will be actually able to look for competitive inhibitors of Calpain using our biosensors. The first experiment I did in that regard is I've shown that both Calpain-1 and Calpain-2 biosensors are responsive to the concentration of calcium. With Calpain-1 biosensor plateauing at low micromolar concentration and Calpain-2 biosensor having a slightly less steep curve. And that was an interesting finding as well, because Also, from literature, we know that there are different calcium requirements for activation of Calpain-1 and Calpain-2, but this suggests that those same differences in calcium requirements for activation appear in the requirements for optimal binding of the peptimains. Next, I expressed this construct, which was based on the large bit Calpain S1 construct, but it didn't have the large bit half of the biosensor, and I used this as a competitive inhibitor of my biosensor. What I'm showing here is that with the increasing concentration of this competitive inhibitor, we are losing the signal, which is just a proof of principle experiment to say that if we are to introduce a competitive inhibitor into our biosensor, we can actually observe that inhibition. Next, I expressed a chimeric protein, which was based on small bit Calpain-2 biosensor, but it had a C-terminus of Calpain-1 peptimain. What I've shown here is that by making that change in the Calpain-2 biosensor, I'm able to increase its strength, its binding affinity, and that goes to show that by making meaningful changes to the primary sequence of our biosensor, we can alter its binding affinity. And having done all of these optimizations and validations, I've done one final characterization of the biosensor, in which I was keeping the large bit concentration constant, but I was changing the concentration of small bit in the assay. And what that allowed me to do was to estimate the dissociation constants of these peptimains in different conditions. So for Calpain-1 biosensor, the dissociation constant was 185 nanomolar. For Calpain-2, the binding was a bit weaker, 508. But then also when we substituted calcium for magnesium, that binding affinity dropped for both biosensors, for Calpain-1 to 360, to Calpain-2 to about 1600. What's noteworthy about this data set is that both of those factors were independently known, is that Calpain's and calcium, and that Calpain-2 binding seems to be somewhat weaker than that of Calpain-1. But to my knowledge, no one's put a number on it before. So that tells us something about the affinity of these peptimains to one another. And in summary, we have developed a biosensor which is very robust, has a very reproducible positive and negative controls, and a very large dynamic range between them. Meaning that we will be able to use this assay to search for inhibitors of Calpain. So we've done the high throughput screening of over 100,000 compounds, looking for compounds that would interfere with our biosensor dimerization. We've done that in collaboration with the Emory University Structural Genomics Consortium and University of North Carolina. And we obtained about 600 hits from that high throughput screening effort. However, when we started whittling down that number by doing validation assays, eventually we ended up just with this one compound, 38A, well, but also with the compound 39A, which was very structurally similar, which even though turned out to be true hits, weren't very good inhibitors of Calpain. In this one Calpain activity assay, 38A was only able to inhibit Calpain by something like 7%, which, again, even though a true hit, is not good enough for our purposes. So even though we were frankly upset by these results, we still wanted to find an inhibitor of Calpain, so we just needed to change our approach. That's when we enlisted help of Dr. Emil Alexov. He is a computational biologist who's specializing on modeling protein-protein interactions. And the approach that he suggested to us is that he would take the structure of Calpain-2 dimer, and he would evaluate what is the effect of each of the residues on the interaction strength of this dimer. So effectively, what he was looking for is for residues that, if substituted, would not change the absolute folding energy much, meaning they wouldn't disrupt the structure of the monomers, but would affect the binding delta G, meaning that those monomers wouldn't bind as strongly to each other. The idea here is that if we were to find those critical residues for binding, then we would know that we could go not for the molecules that just bind anywhere in the Calpain structure, but they would sequester exactly those residues that are important for dimerization. So from here, we had to validate these findings from Dr. Alaxo, who made us this ranked list of important residues. First, I decided to validate these two residues, D812 and Q963. The numbering is not based on primary sequence of calpains, but on the crystal structure that Dr. Alaxo used. And what I've done is that I've introduced mutations at those residues in my Calpain 2 biosensor. And I've shown that these mutations can successfully disrupt the interaction between my biosensor halves to some extent. Doing this side-directed mutagenesis turned out to be very time-consuming. So at that point, I somewhat changed the strategy. At this point, both we and Dr. Alaxo have largely concluded that the C-terminus of CAPN-S1 is the area that's the most important for dimerization of CAPN-S1 with either CAPN1 or CAPN2. And so I decided, instead of doing single amino acid substitutions, I started doing truncations from the C-terminus at these three residues, N957, 957, K948, M924. And what I've shown is that with truncations at these two sites, they, similarly to single amino acid substitutions, are able to knock down the binding activity of this mutant CAPN-S1 to some extent. But most prominently, with the stop code on it, M924, that completely abolishes the binding activity of CAPN-S1 to CAPN1 or CAPN2. Separately, I validated those single site mutations in a live cell model. The way that worked is that I started with CAPN-S1 knockout cells, and then I introduced either wild-type CAPN-S1 or mutant CAPN-S1 into it, and I evaluated how good it was at recovering the expression of endogenous CAPN2, but also how good it was at making an actual active Calpain 2 enzyme. And here we see that this is the quantification of the data. There was some effect on the stability of CAPN2, but depending on which mutation you look at, there was a prominent effect on the activity of that enzyme. So after we've done these few validation steps for Dr. Alaxo, he proceeded to do in silico modeling of over 3 million compounds for us. And at the end, he provided us with 23 hits, which are chemical structures that he believes are most likely to be good inhibitors of Calpain protein-protein interaction. Right now, we've purchased those compounds and we're waiting to receive them. And we will test and validate those compounds first in the nanoluciferase biosensor assay. Then, if and when we confirm the compounds that do have inhibitor activity there, we will test them in the Calpain activity assay. And everything going well, then we'll move on to live cell assay, testing in vitro migration, and fingers crossed, we'll see their effect on in vivo metastasis. That's all from me for today. Thank you to Dr. Peter Greer and everybody else who supported me in this project. And I think we have time for questions, suggestions, complaints. If any. Yeah, mostly, I don't know, because we haven't done exploratory studies beyond this. But what's most likely happening is Calpain has to do with formation of invadopodia, which then, once formed, their apex start releasing metalloproteases. And that's what's actually degrading the extracellular matrix. So that is not to say that Calpain doesn't degrade DQ collagen, but that those cells are unable to form structures that would. Yeah. Compounds. In silico. That's right. Uh-huh.
Yeah. So are those 20
Is 7 the exact number?
Yes. Are those 23 hits like a save? Because I'm assuming there's a lot of redundancy.
I don't know. Are we allowed to show that? I don't know. So they're all polycycles. So they're in a small molecule range, but on the larger side. And the reason why there's 23 hits is not so much that those are the only 23 that seem to inhibit Calpain, but the way Dr. Alexov conducted this in silico assay is that he would model the interaction of the molecules with these several pockets on capaneson, and then estimate what the delta G of that interaction would be. And so it's not that there is only 23 hits, but these are the 23 on top of that ranked list. There's many more compounds than that, but what they did after that is actually do an atomic simulations of how these molecules would come in and bind to capaneson and disrupt the interaction. And what he's telling us is that that process is very time-consuming and difficult. And to date, they have tested and validated in that fashion 23 compounds, and that's the list they sent us, and that's the list we're going to test.
That's a good point.
By eye, not really, but you know, they're all polycycles, and you know, yeah.
I would say that's kind of cool because then instead of family, just like bromine there or whatnot, you can actually do a lot of medicinal chemistry on each of those.
Hopefully.
And create the whole bunch of derivatives.
So that's opening. There you go. Next question. So that's open. Yeah, that's opening another can of worms because, at least to my knowledge, the biggest promise in the field of RAS, for example, is PROTAC inhibitors, which are compounds that, long story short, lead to the protein getting ubiquitinated and degraded. And that's actually one of the approaches that we are also testing in parallel to just, you know, in silico screen described here. I don't have much to show for that yet, though. Whether Calpains could be undruggable, in terms of active site, they're not, because, well, there are known inhibitors, it's just that it's hard to make them specific. With RAS, the issue is that there isn't much molecular surface to bind to, which is not the issue with Calpain, because that's 80 kilodaltons that you can, you know, just pick and choose where you want to bind to. And, yeah, fair enough. In terms of interface where a small subunit binds to large subunit, we know that that contains a lot of hydrophobic residues. And just notionally speaking, that's a place where small molecules would, quote unquote, like to bind. So in that sense, I think we should be good in that we should be able to come up with compounds that will bind there. And if they bind there, they will likely provide steric hindrance and cause the disruption of the dimer. So I think it should be druggable. Yeah, so that's the way that we're trying to approach that PROTAC type pharmaceutical. We actually are trying to do something similar to what people did with that one compound that I showed before, PCB1B. So here, that's the primary sequence that we know that binds to the active site of Calpain but doesn't get cleaved. And here it's conjugated to a penetratin, which pretty much is just a sequence that makes that peptide go through the cell membrane and be better at interacting with Calpain in there. What we're trying to do is to take a portion of this peptide, or a peptide very homologous to it, and actually conjugate it to another small molecule that would be recruiting an E3 ligase. And it's binding in the active site, but it's not per se interacting with those active site residues. That's right. Right, that's a peptide, yeah. Peptidomimetic. Okay, sorry, could you say the I don't know if any peptidomimetics that are targeted against the protein-protein interaction in calpains. I only know of those the inactive site. Yeah, that's right.
Well, it's a pseudo substrate.
I've also tried another thing that I tried in that regard, which perhaps would be closer to what you mean, is that we also did a peptide array screen of looking for peptides that are the strongest binders to Calpain S1 in the primary sequence of Calpain 1 or Calpain 2. And what we have found is, if we can wait for this to rotate, is that this alpha helix is the one that is the strongest binder, at least in terms of primary sequences present in Calpain large subunit to the Calpain S1. I've tested that as an inhibitor in that we synthesized that peptide and tried to, just spike it into the biosensor and see what it does, and it didn't do anything. That's about where it fizzled out. Whether that's the issue with the structure of that peptide on its own or whatnot, I don't know, but that was the extent of it. Did it both ways? Yeah. Yeah, did it both ways, and yeah, didn't seem too promising, at least. Yeah. The answer is yes. That's right. Not to say we didn't lose a lot. That's about 50% inhibition, but fair enough. Mm-hmm. Uh-huh. Mm-hmm. Mm-hmm.
And well, I don't know, separate. Confirmation might not.
Yeah, that's another complication because pretty much nowhere here I looked at the monomer of Calpain S1. Yeah, that's another complication because we had this similar conversation before, and pretty much the reason why I didn't look at the Calpain S1 homodimerization is just because it largely hasn't been physiologically relevant to us in terms of trying to inhibit the enzyme. But yeah, it's possible to look at it. It's doable.
Curious about. Would it be functional?
Thank you.
One of the reasons I know what those bind motion and if it's such a tight interaction, there might not be much breathing going on, and you need to get some breathing to go on in order for small molecules presumably to get in there and block it. So it's not nanomolar, it's half micromolar type of disease, so it is workable.
I think you must win some award for the amount of data shown in the 7 graduations. I had two questions, but the first one was really about what can you learn from Calpain, and you kind of addressed that a little bit. Yes, I mean that's presumably very effective. So what can you take from that would make you learn something better for what you're achieving in that? So that's the question. So Calpain, you showed us earlier.
Yeah. So a little bit of a background on Calpain is that it's an endogenous protein inhibitor of Calpain. And the way it works is that it's largely unstructured on its own, but in presence of Calpain, it sort of loops around the Calpain in that one of the Calpain domains binds somewhere around here where that small molecule is. Then it loops around and goes through the active site. Oh, sorry, you're not seeing this. There we go. Yeah, so one of the domains binds roughly here where that small molecule is, another one binds over here, and it loops around by the active site and actually goes through the active site cleft. But the nature of the primary sequence, it seems, of Calpain is such that it has a very convenient kink, which keeps it away from the active site cysteine, exactly where it would need to be cleaved. And that's the mode of its inhibition, that it just comes up the mechanism, so to speak. So what we can learn from that is that we know the exact sequence of a peptide which has high affinity to binding to the active site cleft. What people have done with that so far is pretty much biochemical assays, that small venture into trying to make that PDB1B exon 1B of Calpain derived molecule. But I don't know of any evidence of that being used in any more complex systems like live cells or animals or anything of a sort. And also, yeah, historically, that sort of peptide inhibition had bad rap in that even if it works well in biochemical assays, doesn't translate well further. And that's why we tried using that peptide in a PROTAC approach, is because those ubiquitin ligase conjugates are known to be good inhibitors, even if those molecules are either bad binders or have poor solubility or things of that nature. But again, I didn't have much data to show on that yet, so yeah, haven't done that yet effectively. Not necessarily. In endogenous protein, that they all bind at the same time, but when you express peptides on their own, I think that's work done. Peter Davies' lab some time ago. The peptide alone is good enough to elicit binding affinity. A very good one too. Wasn't it something like 500 picomolar or something? Something like that, yeah.
That's the way I know for the process. I find two places where molecules are binding and like and protects and what things have gone.
The other question around is expansion sensitivity in this protein. So I'm assuming that it comes back to some structural change. So is that going to be a challenge for inhibition? I mean, there are different levels of intracellular calcium in different locations. That presumably makes this more or less accessible for inhibitory activity. And you showed us, this one slide here from the top left, I guess. So is that telling us that this is going to be very much something that sense this over a wide range of calcium concentrations, but in terms of actually developing an inhibitor that works over that range?
So what we know is most of the time in most of the cell, calcium concentration is at about 100 nanomolar. That's nowhere near enough to activate calpains, either 1 or 2. And it's hard to show that in a live cell in terms of calcium spikes and whatnot, but the working model is that you get these puffs of calcium from endoplasmic reticulum or from outside the cell, and that locally activates Calpain in there. So that also suggests that probably in most of the cell, most of the time, 99% of Calpain molecules are inactive. At least that's what we suspect. So in that sense, if we just work with the structure of inactive Calpain, that's probably good enough. But also another part of it is maybe that's more of a concern if you're trying to go for something that will just change the structure of Calpain. What we hope will happen is if we disrupt that dimerization sufficiently, that will kick in that process of Calpain 1 and Calpain 2 being degraded as in an unfolded protein response, and then you just don't have to worry about it anymore. Too early to say, but that's what we hope for.
Jen?
Maybe Dr. Nicholas.
Oh, he has a hand raised. I think the audio is on.
Can you hear us, please?
Hello?
So,
Is that me? Okay, thanks. It was cutting out a bit. Ivan, nice talk. I had a question about your metastatic data.
Yep.
Do you mind going to that slide, please? Yeah, thank you. So as I understood it, you reported this as sort of standard metastatic burden, and you showed us later some nice images of your lungs. But as you know, the 231 model will metastasize to other organs. And I was wondering if the way that you reported your metastatic burden, does that factor in metastasis to all tissues? Because I know you commented Calpain can have different effects depending on, you know, what players and what signaling partners it may have and expressed in the vicinity. So I was just curious if you'd noted whether there were changes in other metastatic tissues from your genetic manipulations.
Thank you. So in our model system, the most metastasis is observed in lungs and in liver. Fairly common, I suppose. I suppose with breast cancer, bones could be another place to look at, or brain. In our model, lungs and liver are the ones that are, you know, producing the most metastasis, if you will. And when we look at the liver metastasis, if you know, in any particular experiment, if say we get like 50 to 100 micromets in the lungs, we get like 5-10 tops observable in the liver. So in that sense, I just haven't been looking at the data because it doesn't have the dynamic range to work with to try to see those differences.
Thank you.
Depends. That's right. So at least from animal studies, we know that it is possible to have a Calpain 1 knockout animal without any major developmental side effects. Calpain 2 knockout, or you know, when you knock out both with Calpain S1, that's embryonically lethal. However, if you flux that allele, allow the mouse to develop, and then knock out Calpain 2, that's fine. With Calpain 1 knockout, it's fine in development, but when the animals start to age, they develop ataxia, and there is a clot dissolving phenotype which exists but is largely insignificant. So in the essence, so that's to say that it seems that if you transiently inhibit Calpain for a week or two or a month or a year, it should be fine. And that what also helps is that we're working in the context of cancer because, you know, if you develop a new drug that causes neuropathy, you know, that's par for the course in a sense. Yeah, that's the short answer to that. Yeah, that's right. You know, you have KRAS inhibitors again. No, not KRAS, but you know, EGFR, whatever. Yeah, those those would all in comparison be much more drastic. Yeah, that's right. Yeah, fair enough. Yeah, the way I just, just imagine this working is that you would inhibit Calpain until the point of resection of the primary tumor. At least that would be the major part of it. Maybe after that too, for, if you don't catch, another, part of the tumor or whatever. But yeah, that's at least how I envision that working is, the person gets diagnosed with breast cancer, is sent home with Calpain pills, Calpain inhibitor pills, and then, a month or two later, when the tumor is resected, that's notionally when you lose the need to inhibit metastasis if there hasn't any occurred, if any hasn't occurred yet. Yeah. Yeah. For what it's worth, unlike in my mouse model, people don't necessarily come in with 100 mets. Well, possible, but the point is that even say just this notional hypothetical that we have a woman who comes in with a breast cancer and it metastasizes, has a single met in the liver or whatever. If it truly is just a single met here or there, then notionally, if we inhibit any further metastasis from the secondary site, many of those can be treated as well, or by resection or by chemotherapeutics or whatever. So even in the case where metastasis is present, notionally Calpain inhibition can still be beneficial. Of course, it's a whole another matter is that if you see one met, how many do you not see? But that issue hasn't been solved to date yet.